Okay, so let's start at the beginning. The title, algae. This is a color. So how did I come up with that? Well, first of all, I started to think, when nobody else was willing to take this particular date, I said, I haven't talked about algae, really, in any of my lectures. A little bit. And I've always looked for different quotes oftentimes in my lectures. Some of you who've heard a lot of my lectures know that I like to do that. And so the very first time I was on this podium in the lecture series, I read a poem that was written by someone who was on the beach looking out on the ocean and pondering what it must be like far away, out to sea, down deep, and looking at it from the shoreline. And I actually invited the folks in the room that night to that was what we wanted to do with these lectures, was to try to engage everybody who maybe didn't get to go out and see some of the things we all saw. Then there was a time where I actually did a talk around a Kermit the Frog song for inspiration. It's not easy being green. And most recently, I think I took a line out of the alma mater of one of the local high schools that basically implied that the Indian River used to be a lot bluer and a lot different than it is today. And we talked about that last year. So I said, this year I'm going to talk about algae because I haven't talked about it. I'm going to have to beef it up. I'm not going to use a poem. I'm not going to use a frog. I got to go with a scientist. So if you're going to go with a scientist, who's the scientist? First scientist's name that would come to your mind. Top scientist, all time. Number one. Good answer, Dennis Hanisak. Wrong. But you will get extra credit. What's that? Nah. Yeah, that's not bad. Galileo. Galileo is very good. Younger, more recent. Einstein, right. What's the epitome of a scientist that equates to genius? You're an Einstein, right? So I said, aha, for inspiration this year, let's go with Einstein. Albert Einstein has a lot of great quotes. And he talks about science, but he talks about religion, he talks about society, he talks about all kinds of things. So he really was a true genius. So I thought, well, let's go with Einstein. You can imagine my surprise when it turns out that this was not the same Einstein that said this quote, but it was the Walter Matthau version of Einstein. And if you've never seen this movie, you need to see IQ, which was an early Meg Ryan and an early Tim Robbins movie. Matthau was in between his two Grumpy Old Men movies, so this is sort of like a counterbalance to that. And he's a sweet scientist, Einstein, taking care of his niece played by Meg Ryan. And at some point, he utters the words, algae, this is a color. But it turns out that actually color of algae is a real important thing historically with how we've looked at these, and that sort of led me to the train of thought that evolved into the talk tonight. So my overview is, first of all, describe to you what algae are in general. I'm going to give you an overview of what I call algal biodiversity. I'm going to talk about why are algae important. And then I'm going to sort of seg into some of the things that I've done over the years that I'm just going to select a few examples to try to support some of the other things I'm saying about algae. And over the years, I've actually worked in a number of different habitats or environments or ecosystems, including coral reefs, deep water algae, and so on. And I decided to take two because of the time limit. And I'm going to talk a little bit about Indian River Lagoon, and I'm going to talk a little bit about marine biomass farming, which was some work that was more in the aquaculture area. So first of all, when I say to you, what do you know about algae, what are you going to tell me? Slime. There it is. I'm waiting for it. Slime. What else? Slimy, scummy, gets worse after that, right? And that, in fact, is what most people think about algae. And I'm going to try to give you the rest of the story here with the time I have. Alga comes from the Latin word for seaweed. Seaweed is a common name for the macroscopic algae. Probably most of you have used that. They all are photosynthetic, producing oxygen, fixing carbon, just like a higher plant would. They all use that master pigment of photosynthesis called chlorophyll A. They have very little tissue differentiation compared to what we consider to be higher plants, so you're familiar with trees and grasses. Algae have no organs. What is an organ in a plant? An organ in a plant is root, stem, leaf. So pretty simple. But algae don't even have anything that complicated. And they've got no vascular tissue like trees do, for example, to move water through the length of the plant. Algae have simple reproductive structures. So unlike, for example, seed plants that you know about or flowering plants, they have no protective cells, extra cells around the zygote. So when they reproduce, it's very simple without a lot of extra protection for the cells. One of my real pet peeves is you use the word algaes. Algaes is not a word. Now, no offense to anybody who uses that, but I won't answer your questions if you use the word algaes in your question. The singular is alga, the plural is algae, okay? There's no such thing as algae. Many, many scientists actually use the word algaes, and I just shake my head and move on. But don't do it. Phycology, I study phycology. I call myself a phycologist. Comes from the Greek word meaning the study of seaweeds. And in fact, again, the emphasis on the word seaweed, that's what the ancient Greeks and Romans had studied. And actually, some of the early classification schemes for organisms were based on where they lived. So if it was land or sea, they were considered different types of organisms. If you Google phycology because you're so excited to hear about phycology, the first thing Google comes back with is, do you mean psychology? I'm not making that up. Go do it. Let's talk about algal biodiversity. But first, I have to tell you, because I realize sometimes that we use these words and not everybody knows what they mean. Biodiversity basically is just the diversity of life. What does that mean? All living things comprise the Earth's biodiversity. And this includes things at the species level, so different types of organisms. It includes things at the molecular level or genetic makeup, and also the natural communities in which they live, so habitats, ecosystems, and so on. The number of algal species is actually unknown. There's something like 125,000 that have been described, but if you look at which ones are synonyms and so on, probably there's about 30,000 that have been legitimately described and probably are valid. So that'll give you some idea of the numbers of species that we have in terms of algae. The first person who actually classified algae at a higher level was the Irish phycologist, William Henry Harvey, who divided the algae into basically 4 groups, primarily on color or pigmentation. And these were known as red algae, brown algae, and green algae because of their pigmentation that masks the chlorophyll A, which is green underneath. Depends on the type of group that you're looking at. So the red algae have different accessory pigments than do the brown algae, than do the green algae. And then the last group was diatoms, which is a microscopic group that had received a lot of attention in the 1800s, and we'll talk about them in a little bit as well. Harvey was the first person to really use biochemical criterion in plant systematics. Nowadays, we use biochemical criteria all the time. We use molecular criteria all the time. He was really the first one to start to do that, and he did it at a higher level than anybody had done before. Nowadays, much more modern approaches, we look at molecular information, and we build, this is part of the tree of life that you can find on the internet. This is an attempt to look at the genomes or the molecular makeup of all organisms and then show their natural relationships. And algae don't fit a single group. I highlighted some of the areas here. All green algae and all higher plants. So every plant that lives on land is up here. All animals, for example, are this one little line here. All animals. The rest of these are where the algae are. So some of these macroscopic algae, the greens and the reds, are up here. The brown algae are down here. The diatoms are down here. A lot of other algae are down lower. Most of all these other groups are all primarily microbial. So one of the things that we oftentimes forget as humans is that there's this incredible microbiota out there. So algae are very heterogeneous in both in their chemistries and in a lot of other ways. I'm going to run you through a very quick overview of some of these different groups to give you an idea of the diversity that they have. The blue-green algae was a group that came after Harvey, and for a long time people would call the groups based on color. These were the blue-green algae. They were the golden brown algae, and so on. And we now know these not to be related at all to the rest of the algae in a very close way. They're considered bacteria, specifically the cyanobacteria. And examples of these include very small coccoid cells that are photosynthetic. Prokaryotic was not known until the mid-1980s, and we now know that maybe half of the primary productivity in the world's oceans might be due to this tiny cell. It's less than a micron, which is a millionth of a meter, and they were missed by oceanographers for a long time because they went through all the nets. We couldn't catch them. We now call these picoplankton, very tiny plankton. One of the important things about this group, as is the case for all bacteria, is that they're prokaryotic. Prokaryotic is a fundamental difference in cell type compared to the rest of the organisms, which are eukaryotic, that you are most familiar with. And prokaryotic means they don't have a traditional nucleus. They have other differences in their nucleic acids, and they have very little other organizational at the cell level, so they don't have organelles. Lyngbia is a filamentous alga, and we find this in the Indian River Lagoon, and we oftentimes see this floating around in the summertime. Also, people have studied these off the coast of Florida, the impact on coral reefs because they produce a lot of toxins. Valerie Paul, who's in the room tonight, who is the head of the Smithsonian down in Fort Pierce, she has done a lot of work on their chemistry. Amy Wright, who is here at 4 o'clock, also has done some work on understanding the chemistry of Lyngbia. And there's many, many different types of toxins and interesting natural products that Lyngbia produces. Anabina is another filamentous one, and this is an example of one that has this special cell here called a heterocyst. Not all blue-green algae have this, but whenever you see that, it means that they're fixing nitrogen, that particular cell. And this is a process that goes back, as you'll see, billions of years ago when the Earth's environment was quite different. And actually, oxygen is toxic to nitrogen fixation. And what's amazing is that, in this case, there's a specialized cell that this process occurs, but there's a lot of these cyanobacteria that can do this without a specialized cell. So these very, very tiny cells, in some cases, are able to fix nitrogen, and they're able to keep the oxygen part of their metabolism separate from their nitrogen fixation part, which is very important because oxygen is toxic to nitrogen fixation. So that's very important. They're the only algae that can provide new sources of nitrogen, so they take that atmospheric nitrogen. Spirogyra is, it's hard to see a little bit maybe, but this is actually a spiral. It's like a coil. If you were to stretch it out, it would be like a spring. And you might know Spirogyra because it's grown commercially. This is actually raceways in Hawaii. And one of the main uses is actually in health tablets. And people will eat these to take some advantage of the nutrient and special ingredients that some of these algae have. Historically, this was eaten as food in certain parts of the world and became more of a health item in our part of the world. Diatoms, I mentioned them earlier as being one of the original groupings in the 1800s. And diatoms are interesting because their taxonomy is all based on the dead cell. And the dead cell is all this silicon or glass cell wall. So think of these as little petri dishes. You know what a petri dish is. And there is a top part of the dish and a bottom part of the dish. And the diatoms are sort of put together in the same way. And diatoms come from the Greek word cut in two, and that refers to these two different parts of the cell, or valves, if you will. Diatoms are very, very important ecologically. They're important as aquaculture feeds. Skeletonema is a well-known diatom that lives in coastal and estuarine waters. It's in the Indian River Lagoon. You can find it throughout the world, up and down the East Coast. There's also some centric diatoms, which have these really nice markings. And the whole taxonomy is based on the details of those markings. In the 1800s, this was actually sort of a hobby where people would place these on a microscope slide and make designs, and then they would send them, out to people as a gift. Some of them have special adaptations like spines. This is Catostris. There's many species of Catostris in our area. And the spines can be useful for flotation and also to discourage grazing by zooplankton. Dinoflagellates. And it is dinoflagellates, not dinoflagellates, although you hear most people say dinoflagellates. Dinoflagellates comes from the Greek word meaning whirling, and flagellum, which is a Latin word for whip. So they're whirling whips. Flagella are like little whip-like appendages that look something like that. dinosaurs, as you know, are terrible lizards. That's what that word means. And so if you said that dinoflagellate, then you would mean terrible flagellates. And in fact, this is a whirling flagellate. Okay? And you may know these as red tides, because some of them produce red tides. And one of them in particular produces the Florida red tide, which is this guy here, Carina brevis. And that's actually named, the genus name is actually named for Karen Steidinger, who has worked for many years over in St. Petersburg on the Florida red tide. Another interesting one that we find in the Indian River Lagoon is this one here, Pyrodinium bahamense. Some of the strains are toxic, and in particular they produce a saxitoxin, which is a neurotoxin, very potentially deadly. And a few years ago, there was incidents where this had been concentrated up in the food chain into pufferfish. People who were eating pufferfish from the Indian River Lagoon got very ill. And so in fact, pufferfish are now banned for consumption in the lagoon because of this potential problem. But dinoflagellates are not all bad. This is Symbiodinium, and you'll hear about corals next week, and you'll hear probably a little bit about coral bleaching, and you've heard about coral bleaching in some of our other lectures. Symbiodinium is a non-flagellated dinoflagellate that lives inside the corals as a symbiont. And its relationship with the coral is very important to the health of the coral. This is why when you swim over a coral reef, they're golden brown. Okay, it's because of the color of the zooxanthellae was the earlier name for this particular group. And zooxanthellae is still sort of generically used to mean those symbiotic organisms. Coccolithophorids are another group of microscopic algae. They're little green guys. They got two flagellates, but they also have something in between that's called a haptonemma. And the group, taxonomically, the haptophytes, was named because of the haptonemma. Haptonemma refers to this little extra flagellum-like structure, and it comes from the Greek meaning touch or fasten and a thread. And what this is believed to be, the function of this is believed to be more in terms of particles, ingesting particles, or also sensing the bottom, things like that. What's amazing about coccolithophorids is they produce coccoliths. That's what coccolithophorid means, bearing coccoliths. And these are all of these calcareous scales that grow external to the cell. So these individual unicellular organisms produce this incredible ornamentation. And when the cells die, they oftentimes will settle to the bottom of the ocean. This is more of an oceanic type of group. And you may know the White Cliffs of Dover. This is a geological accumulation of coccolithophorids. This is all of these guys, fossilized. Okay, so that's a rather impressive thing to see. Macroscopic algae now, the red, brown, and green algae. The red algae are named because of their rose and plant. Whenever you see phyta, it always means plant. Rhoda means rose or red. So in the Indian River Lagoon, we'll talk a little bit later about Gracilaria tigvahii. That's probably the most abundant one in the Indian River Lagoon. Maybe you walk along the beach or you snorkel out to the worm reef. You might see this beautiful alga, Botriocladia, which has these vesicles that look like little pearls underwater. They're important red algae that are calcified, and these are important in coral reef. So this is Neogonolithon, and think of these as sort of the organisms that cement together the different parts of the reef. So it's not just coral that produce that structure. There's a lot of coral and algae that also contribute to it. And these algae are also very important for settlement of coral larvae. They will preferentially settle when there's an algal like this out there. So they're very important for coral reefs. And here's another one that is more upright. We call that an articulated corallin. But that's Jania. We actually have Jania locally in the Indian River Lagoon. It's quite an interesting story, which I won't go into now. A lot of red algae are economically important. Some that come to mind to me right away is Porphyra. There's lots of species of Porphyra, but the really good ones are cultivated in Japan, and they make these dried sheets of nori. And you probably know nori because it's the wrapper on it's the outer part of your sushi. So if you like sushi, what you're eating is various things that are wrapped with nori. Okay? And they're grown, they've been grown since the 1600s on basically ropes and nets like this, And again, there's quite an interesting story as to how that all happened. Another one is eucheuma. This is grown more for a commercial product. There's a whole category of compounds that come from algae called phycocolloids. And eucheuma is a source of carrageenan. Carrageenan is one of the key ingredients in ice cream, for example. And these are grown most predominantly now in the Philippines. And they're often harvested by fishermen, and oftentimes it's an alternative source of revenue for them. And again, they will be cultivated on rafts or lines of some sort. Gracilaria is another one that has been grown commercially in various places in the world. In the 80s, there was a big increase in Gracilaria production in Chile, and then more recently, it's been in Indonesia. And again, here's an example of what it looks like in Indonesia. Again, this is the type of thing where it's open water, usually very shallow coastal waters. It's labor intensive, but the labor source is very cheap. Oftentimes, again, it's secondary revenue. Oftentimes, it's like family of fishermen that will do the work, and they sell it to the phycocolloid companies. Let's talk about green algae a little bit. Green algae means green plant, and most green algae are actually tiny, microscopic forms. Most of them are freshwater forms. This is Chlamydomonas. Chlamydomonas is like a white rat in the phycology world. It's used tremendously for plant physiology studies because it being a green alga, it has the exact same pigment and the exact same biochemistry as do higher plants. So it's a lot easier to do physiological work with a culture of Chlamydomonas than it is to, say, do it with a larger tree or a corn plant or things like that. So a lot of basic physiology that we know about plants that have been modified for, say, agriculture derives from work on Chlamydomonas. So it's quite an important plant. I was thinking when I was putting this talk together, it was the first time I ever saw an alga. I decided it was in high school when I was taking my biology class, and I think it was probably Spirogyra. And I've always liked Spirogyra. I don't know if that's the reason or not, but it has a spiral chloroplast. That's where all the pigment is. And so it's very easy to tell the genus of that particular group. So maybe you've seen that in your biology classes. And again, as I mentioned earlier, these were all ancestors to land plants. So the evolution of terrestrial plants came from green algae, and that was a very important event indeed on the planet, which we'll talk about in a little bit. So some of the bigger forms, and some of the ones you may have seen, you may know sea lettuce, called Ulva lactuca, and that is grown locally in the Lagoon, and I'll talk a little bit more about it later. The plant that I chose to work on for my PhD work in Rhode Island was this one, Codium fragile, which actually was an invasive species, and it was back when we didn't know much about invasive species and what was important about their characteristics that made them invasive. And this is one of the really first algae that we knew to be an invader throughout the world. And it was first introduced in Long Island Sound and then spread up and down the coast. And one of its common names is oyster thief, because it would grow on oysters, and after it got really big and buoyant, it would just pop the oyster off the bottom and float the oyster out to sea and onto a beach. More recently, this is an invasive species in Florida. Maybe you heard Brian LaPointe lecture about Caulerpa brachypus. This is a species that is known primarily from the Pacific. We don't really know how it got here off the coast of Florida. Probably aquarium trade, but we don't really know that for sure. And in between the time this appeared in 2002 or so, in the early 90s, we had quite a large amount of Codium ismocladum, a different Codium species growing in that same area. So Brian and I were both working on that, and then he's basically done it for 20 years since then. There's also calcified green algae, such as Halimeda. This is very common in the Florida Keys. We have some locally, but it's much more dominant in the tropics. Halimeda is important because if you look at the sands on the beaches in, say, Florida and the Caribbean, most of that's calcified material that's been produced by organisms that has been ground up, and the highest percentage of that comes from this alga, Halimeda, this genus. If you go to a place like Florida Bay where there's a lot of fine sediments, more mud-like, they are actually derived from a lot of the calcified algae such as Penicillus. So some of these algae are just very important geologically and also in terms of sediment cycles. Let's talk briefly about the brown algae. Locally, the ones we probably know the most about, the ones that you probably have seen, is the sargassum that's cast ashore, or maybe you go out fishing and you know that if you go out and find the weed lines, the sargassum weed lines, you know that's where the fish are. And maybe you've actually gone to the Sargasso Sea. I did that once. That was a big treat for me. The Sargasso Sea is actually named for the plant, sargassum, for the alga sargassum, which was named by the Portuguese sailors after a species of rock rose called the same name, sargasso, in Portuguese. The story goes that Christopher Columbus in 1492 was going across, and the troops were getting a little restless. They wanted to mutiny, and they were in the Sargasso Sea, and they started to see patches of this. They named it for something that reminded them of home, and I guess they continued on to discover what became the New World. In the Indian River Lagoon, the largest seaweed we have is a sargassum, Sargassum filopendulum, and it grows attached, and it grows quite locally here to Harbor Branch. If you go to the Florida Keys, you oftentimes hear algae overgrowing the reef. Generally, we're talking about Halimeda, but also this brown seaweed, Dictyota. It's very, very common, very important. Also has very interesting chemistry, which Val Paul can tell you about. A large group of brown algae that we do not have here is the kelps, but you guys have been various places in your life, and I'm sure that you have seen kelps if you are in more temperate waters. Laminaria is the genus name. That is found in New England and actually all throughout the northern part of the world. You may know Macrocystis. It grows in various places in the world, but off of California, it can be 200 feet long, 65 meters. Largest plant, largest alga in the world. Maybe you've gone to the Monterey Bay Aquarium. If you haven't, you should, because this is the best place to see algae where you don't have to get wet. And they do a wonderful job not only displaying natural populations of Macrocystis, but they probably have 60 or 100 species that you can go around and look at on the algae. So I love the Monterey Bay Aquarium. Maybe you know that for many years until very recently, there was a whole industry off of California. This is a boat harvesting Macrocystis. And that was harvested for a number of reasons over the years, most of the time for alginates. Alginate is another phycocolloid. Alginate has a lot of uses. It's, for example, in a lot of food products. Maybe you know alginates because if you ever had a dental impression and they threw something in your mouth, out came a mold, voila, that was alginate. Okay. In Japan and China in particular, they grow Laminaria japonica, which the name there is kombu. And this is the leading fisheries product really in the world. And this is primarily totally for food. And there's a whole other side of seaweeds that we don't see in the United States. And that has to do with the food side of it for human food consumption. And that's mostly entirely really in the Orient. Next thing I want to point out is that algae have been here a really long time. And I want to take you back through the fossil record. The very oldest fossils that we know on Earth from 3.9 billion years ago were probably the first cyanobacteria. About 500 million years later, so it took a while, we have fossils of the first oxygen-producing cyanobacteria. And I said how important that was because it was a very different environment. There was no oxygen in the air. It was a very reducing type of place. Take half that time from 3.4 till now, and it took another 1.8 billion years before the first multicellular algae were found in the fossil. There's evidence in the fossil record. These are all red algae and green algae, so they are the oldest macroscopic algae. Much more recently, almost 500 million years ago, we started to see the other group of brown algae. Remember I said earlier that molecularly we now know they're very, very different than the other two. And that was a much more recent event. And then about 360 million years ago, we got those coccolithophorids, and I mentioned the White Cliffs of Dover. They're those organisms. And about 200 million years ago, the first Dinoflagellates, and then much, much more recently, the first Diatoms. So this has taken quite a long time to get this great diversity of algae. And this is just a very quick plot through time. This is like 4.5 million years ago. So the first algae were out here, and you can see that this is oxygen relative to now. So the 100% here is what our oxygen level is now. And you can see that for most of the life on Earth history, the oxygen levels were much, much lower. And then when the cyanobacteria started to produce oxygen, at some point it reached some threshold and it really started to spike. Land plants, so these little simple algae got about half or 60% of the oxygen level up. And then when land plants formed on land, that really kicked up the oxygen level, and now it's been stable for several hundred millions of years. So this is kind of interesting. But it shows the relationship of our global environment to these organisms, and the fact that something that happened maybe a billion years before led to conditions that allowed some of these other things like multicellular organisms, which without an oxygen environment could not exist. So this is actually kind of really important, I think, to hook the fossil record with what happened with oxygen levels. Algae are important for many reasons. I've listed them here, and I've mentioned a lot of these in my examples earlier, so I'm not going to go through these in great detail. I'm going to pick one later. Also, I've mentioned a lot of the economic uses of algae, so I'm not going to go through them. But I will mention two others. One is fisheries. There's a very important relationship of algal habitat and algal food supply to fisheries. And think of the macrocystis forest off of California and the relationship it has to the fish that say live in that habitat. Another thing I think is again using the kelp idea, recreation. I mean, sometimes these are just great places to go. I mean, if you've never gone scuba diving in a giant kelp forest, you should do that if you can. It's really quite cool. And it's hard to put dollars on some of those. So let me now talk a little bit about some of the reasons why algae and plants in general are different than animals, and why they do things that we may not think about right away. First of all, here's a little chemistry, a little equation for photosynthesis. This is really one of the most important things that happen on Earth. Plants, including algae, in the presence of light, so photons of energy from the sun, and the presence of that master pigment, chlorophyll A, can fix carbon dioxide from the atmosphere, from the water, and produce sugar. This is glucose, but produce sugar, and then give off oxygen. So they're fixing carbon dioxide, they're giving off oxygen, they're making food. I may talk about productivity when I talk about this process. That's the rate at which organic matter is produced. I might talk about production, which is the total amount of organic matter that's produced. So productivity is more a rate per unit time per unit area. Production is more like the total amount. And algae, just like plants, can take up inorganic nutrients directly from the environment. So what does that mean? Well, they can take up nitrogen from the environment as ammonium or nitrate, and they can take a sugar molecule, they can add an ammonium ion to it, they can make amino acids, and then they put the amino acids together to make the proteins. So they do this all from scratch, so to speak. They take the energy from the sun, they take the inorganic elements from their environment, and they make biomass. They make living material. Animals require that to be existing already. So animals eat things. Okay, so animals don't have that capability to directly make energy from the sun or to take up inorganic elements. That's what plants do, and algae. So one of the recurring themes is that energy flows. So energy from the sun going into that primary productivity level, such as algae, being consumed, say if it's a phytoplankton, being consumed by zooplankton, being consumed by a small fish, being consumed by a bigger fish. At each step, there's a loss of energy. Energy's flowing through the system, but nutrients are cycling. So when those plants take up the nutrients, when they die, they're going to release the nutrients. The animals will take up the plants and they'll excrete nutrients. So nutrients are constantly cycling in the system. And I'll bring these points up later. So I want to give you one concrete example. And the inspiration for this was, I was trying to think, well, maybe how could I explain some of these terms, some of these ideas to you all to understand how little we really understand the importance of algae. And a couple years ago, I had a colleague at a meeting give a talk, and he presented the basic concept here that I'm presenting. And his name is Bob Anderson. And I talked to Bob recently electronically, and he said, yeah, go for it. So I'm presenting some of this based on his inspiration to me. And it basically starts off with the premise of how many cells of phytoplankton, these little algae, are in the oceans at any one time. Now, I sort of tried to ground truth this today and yesterday, and I found numbers all over from 10 to the 23rd to 10 to the 29th cells in the oceans at any one time. Those are huge numbers. I kind of took the average, or the most commonly cited numbers, and that turns out to be about 1 times 10 to the 24 cells. I don't know that we have a name for how many powers that is. It's long past any zillions of numbers that I know. But 10 to the 24 cells. They're very tiny, though. They're only 2 microns, or micrometers, average diameter, because most of them are small out in the ocean. And that's about 0.0008 of an inch. So really small. And these are basically bacteria size, or a little bit larger on average. So the question is, if you've got 1 times 10 to the 24 cells, they're all 2 microns wide on average. If you put them end to end, how long of a line would that be? Anybody want to guess? I know you got an answer. All right, I'll tell you. It would go from the Earth to the moon and back. Pretty incredible, right? But it doesn't do that once. Doesn't do that twice. Doesn't do it 3 times. It does it 1.5 billion times. Now, that's really hard to imagine. I can't imagine it. It's hard to imagine a magnitude of this number. It's hard for me to imagine how small these cells are. It's hard to imagine how many times back and forth from here to the moon. So, maybe we can do this a different way. We can arrange the cells into 2 dimensions. And so we just pack the cells together like that. And we can make this, say, 12 inches wide and 3 inches deep. So it's a piece of wood. Think of it as a piece of wood, 12 inches long, 3 inches deep, something like this. How long would that piece of wood be? How long would that piece of, it's not wood, but how long would that piece of algae be? A plank of algae. Anybody want to guess? It would go from here to the moon. So imagine that. So you see, we could actually build this, and we could actually do the walk to the moon. But that's incredible when you think about that. Now, here's what's even more incredible in my mind, is that on average, phytoplankton are dividing once per day. So that means they're doubling every day. So what does that mean? That means you'd have 2 boards tomorrow, okay? But we don't have 2 boards tomorrow. Why is that? Because one's being eaten every day. It's eaten every day by protozoa, little animals, microscopic animals, zooplankton, fishes. You know, sometimes whales will feed on phytoplankton. So this daily board, if you will, of phytoplankton is what feeds the ocean. And if you eat seafood, or if you get some revenue from the sea because of marine organisms, you're depending on that board of phytoplankton. So I thought that was a way to try to relate it. The point that algae provide food for animals in the estuaries and the oceans. And in so doing, I showed you the equation that they fix carbon dioxide and they produce oxygen. And I showed you that through geological time. That's why we got an oxygen environment instead of a toxic atmosphere that none of us could live in. But let's talk about today. Okay, every time I take a breath, 50%, half of them, that one's from the land. And my other breath, out of those 2, the other 50% is from the oceans, from the algae. Okay? Second one was a little saltier, by the way. That's amazing when you think about it. You know, think about that. Every time you breathe, where'd that oxygen come from? It came from the plants. Another way to do that, of course, would be you could do all your 12 hours of breathing, you know, on the terrestrial plants, and then you'd skip the algae for 12 hours, so you couldn't breathe for 12 hours. But that, I didn't want to demonstrate that. So the oxygen in every breath we breathe comes from algae. So therefore, without algae, we would not be here, even in terms of the long scale, evolutionary-wise, or in terms of our more daily life. So I want to phase into the second part of my talk a little bit about some research stuff. Indian River Lagoon, a lot of you know about this. I've talked a little bit about the lagoon in the past, but I know a lot of you live right here, and you know about the Indian River Lagoon. The Indian River Lagoon is really special. It's one of the longest lagoons in the continental United States. It's about 40% of the east coast of Florida. The Indian River Lagoon is often called the most biologically diverse estuary in North America. This is due to its large numbers of species, a favorable climate, and we get species that come from more tropical waters to the south and more temperate waters to the north. And the lagoon is just an incredible fertile mixing bowl of organisms, of life. You also know that we're living in an area that is rapidly being urbanized, and so it's a challenge to have such a productive natural system that we may potentially be impacting. If you care more about the dollars, the economic impact of the Indian River Lagoon in the last year or so was calculated to be more than $3.5 billion per year to Florida, so that's every year, $3.5 billion. In the early 90s, the water management districts and other agencies decided that a high priority for maintaining the biodiversity in the Indian River Lagoon is to keep it a seagrass-based ecosystem. And why is that? Bottom line, those of you who heard me talk before, the more seagrass you have, the more fish you have, because it's great fish habitat and it's great for major invertebrates as well. So I was always interested when I first came to Harbor Branch how little research was done locally on the Indian River Lagoon marine algae. And a lot of the literature was old, outdated. And honestly, it was quite a challenge to work on the macroalgae of the Indian River Lagoon. But some of you may have heard a talk I gave in 2008, I think, with Mark and Diane Littler. They were the first authors. Diane was the first author, and then Mark and myself on this book, The Submerged Plants of the Indian River Lagoon. And it's probably one of the funnest things I've ever done in phycology because of them. And to me, this just says the tip of the iceberg about biodiversity and about algal biodiversity. We recorded 229 species of marine algae, which was a lot more than anybody thought was in the Indian River Lagoon. But what really strikes me as amazing was that more than half of those, 57%, were new records for the lagoon. Now, this lagoon is right outside our doorway. The average depth is 5 feet, 1.5 meters. We've lived here for a long time, and yet in 2008 we can go out and decide that we have all these new records for lagoon. 20% were new records for Florida, and 1% was new records for the Western Atlantic. So to me, that just tells me, what an exciting place the lagoon is. If you're interested in the breakdown, the flora, so to speak, is dominated by red algae, 112 species, and then green algae, and then brown algae. That's very typical of warm water and tropical waters in the world. Most of the algae, macroalgae in the lagoon, is so-called drift algae. And that's because there really isn't much hard substrate in the lagoon. As you know, it's soft sediment. And there's not a lot of places for the algae to attach, which is what you see when there's rocky areas. And so the algae actually does move around based on winds and currents. And it's actually very hard to know how much of it is being produced in one location versus how much is drifting in from other locations. This is a picture of Sherry Reed. Sherry Reed was at 4 o'clock lecture, and she was a huge help in this project, and just really loved working with her too. So I got a picture of Sherry. And what we were really interested in is being able to identify some of these drift algae, which before this was incredibly hard to identify. And if you go back through the literature, you don't really know what anybody is talking about when they refer to some of the algae that's been reported in the lagoon. So we now have this book, and many of you have seen it, and hopefully I think it's going to really trigger a lot of additional work on the macroalgal community. It's helped me because I'm very interested in things like, well, how much macroalgae is actually in the Indian River Lagoon? So I can tell you that I know it's highly variable in time and space. And I have consulted with Lori Morris at the St. Johns River Water Management District. Lori and I have worked together periodically over the years. And again, another wonderful person who works in the lagoon. And she has done some estimates with some of her colleagues that if you take the Indian River Lagoon segments south to Indian River County, so not the whole lagoon, and she did this for 2 years, she got about 70, let's call it 80,000 metric tons. That's a lot of algae. I did an extrapolation, so if it's wrong, it's all on my shoulders. But taking the area of that study and the entire area of the Indian River Lagoon system, my estimate is that there's probably 170,000 metric tons of algae in the lagoon. That's quite a lot. So one of the obvious questions is, how does that tie into, say, nutrient loading into the lagoon? And in total, the lagoon receives quite a lot of nitrogen and phosphorus. Most of this comes in from discharges from freshwater sources, the canals that we all know about and some of us live on. And in this paper, which again is hooked to the Water Management District, there was this comment about that the current rate of nutrient loading is causing a shift in the primary producers of the Indian River Lagoon from macrophyte, meaning seagrasses, to phytoplankton or algal-based system. And so there's some concern raised about that. Now, how true that is, I don't really know, but it's certainly a question I'd like to answer. And in particular, I'm interested in is the growth of macroalgae in the Indian River Lagoon limited by nitrogen? And does macroalgal growth in the lagoon then respond to increases in water column nitrogen? So I work mostly in the local area from Vero Beach south to Fort Pierce, and the Fort Pierce Inlet's down here. And we've had a number of stations in that area for a couple of years. And if you're interested in the overall patterns from those discharge points in Vero Beach as you go from north to south, this is the amount of nitrogen. You can see it decreases through time. And the first year was a wet year when we had a lot of rain and a lot of discharges. The second year was a beginning of a drought year. And you can see that it does matter how much nutrients are discharged depending on the weather and the discharge rate. And you can see that for phosphorus, perhaps it's even more dramatic. So this is nitrogen, that's phosphorus. Those are the two main nutrients when we talk about algal growth. Over the years, I've been very interested in using algae like biomonitors and looking at the tissue nutrients. And this is productivity, so that's the rate of primary production, grams per meter square per day. And you can see this is internal nitrogen, what's actually in the plants. Initially, when you increase the nitrogen in the plants, it increases its growth rate really fast, very explosively, and then it levels off because it's got as much as it needs. So think of this like when you fertilize your lawn. You know that you need to fertilize a certain amount. If you add too much fertilizer, it doesn't do any good. It's actually waste, and actually it's probably not an environmentally good thing to do. So I've used that over the years, and I've done some experiments like this one where I've looked at the productivity of things growing here in the Indian River Lagoon, and they grow very fast. They start to slow down, and you look at the tissue content of nitrogen, you see that's because the nitrogen level now gets below this magic 2% number, this critical level, and it starts to go slower and slower. If you add a little spike of nitrogen here, that's what this arrow is, you can see that the internal nitrogen very rapidly increases, as does growth rate. And you can repeat this on, and if you do a control setup where you add no nitrogen and just let it grow, you can see it just sort of slowly continues to grow slower and slower and slower, and eventually just sort of zero out. So we've looked at this through the year, and we see this. This is that 2% number for tissue culture. If it's above this number, that means it has plenty of nitrogen. If it's below the 2% level, it means it doesn't have enough nitrogen. It's hungry for nitrogen, starved, if you will. So you can see that in the dry season, that's when you have the lowest tissue nutrients. That's when nitrogen is most likely to be limiting. And then in the wet season, after we've had extensive discharges, that's when you have the maximum amount of nitrogen. So it does seem that it tracks it very well. North to south, this is what it looks like. So this is Vero Beach up here. This is where Harbor Branch is. So Harbor Branch is right about that magic 2% number. And then closer to the inlet, you can see there's probably nitrogen limitation. So the answer to the question is it depends where you are and when you're there. So from north to south, nutrients decrease. And pulses of inorganic nitrogen are usually associated with the initial onset of the wet season. And if we have tropical storms or hurricanes or any kind of major discharge, we see these pulses of inorganic nitrogen. The algae are able to take them up very rapidly, and the tissue nitrogen reflects what's in the environment. And we've done studies that we have looked at the growth rate as a function of the nitrogen content, and we see the relationship that I mentioned earlier. So nitrogen limitation does occur both spatially and temporally in this segment of the Indian River Lagoon. And so what I think this means is that there's a lot of proposals out there to reduce the amount of freshwater discharges in the lagoon to reduce nutrient loading. There's going to be an EPA meeting here at the end of the month where they're going to try to set targets for the Indian River Lagoon. That'll be very interesting. And I think that if we are able to reduce those nitrogen loadings, we should see a decline in macroalgal productivity and abundance, and it should lead to reduced competition of macroalgae with the seagrasses in the lagoon, so the concern that was raised earlier. And then very briefly, I want to talk about algae for biofuel, because actually that's why I first came to Harbor Branch. I didn't mention that. But I was going to solve the world's energy crisis about a while back. How are we doing? Still working. Okay. Remember that formula for photosynthesis, very important. Plants have this incredible capability, and algae have this incredible capability to take light from the sun and make biomass. And remember fossil fuels. That's what we all use. To get here today, how'd you get here? You used fossil fuel, right? That's how I got here. Fossil fuels are formed by anaerobic decomposition of organisms, especially phytoplankton and zooplankton, usually in deep anoxic seas or lakes. And this was done millions of years ago. Fossil fuels are not renewable unless you think that you can wait millions of years for it to happen again. So they're not renewable. This is addressed to the baby boomers in the crowd, especially me. I'm a baby boomer. I find this a sobering statistic. In one generation, if you were born in 1950, and the average life expectancy for a baby boomer in 1950 is 75 years, so 2025. During that period of your one life, that one generation, 70% of all U.S. oil will have been consumed. You can see it was much lower before that generation, and it's going to be much lower in the future. That's amazing. So basically, one generation of Americans took and used 70% of all the oil that we have. Okay, that's incredible. So, could algae be used for biofuel production now? Don't wait a million years. If the algae can do this marvelous thing, why can't we capitalize on it now? And in fact, I said this is why I came to Harvard Branch in the first place, and I came because John Ryder had started some projects here in Florida at Harvard Branch. Previously, in 1969, he started what he called at the time a marine aquaculture tertiary sewage system complex. And the whole idea here was to take raw sewage or secondarily treated sewage, grow phytoplankton. Algae love nutrients. I've already talked to you about that. The best thing you can do with phytoplankton is feed them to a filter feeder, like a shellfish, like a good old clam. And what he found was that the phytoplankton could take up a lot of the nutrients. That was a good thing, because back then we were still discharging sewage out quite a lot into the marine environment, much more so than we are now. And he found that the shellfish readily consumed and grew quite well on these phytoplankton. But the problem was they're animals, so they excreted all their waste. Remember, nutrient cycle, right? Energy flows, nutrient cycle. So they excreted everything back out, and he started to use seaweeds as the last step. He called it a polishing step. And the idea here was to strip out all of the nutrients from the shellfish. Well, it didn't take long for people to think, hey, the seaweeds are growing incredibly fast. We had no idea seaweeds could grow this fast. They're taking up all of the nutrients. Why bother with all the in-between steps? Can't we just go from sewage to seaweed? And in fact, that's one of the first things we did here at Harvard Branch. And the answer is yes. And this is a time when we had just gone through oil embargoes in the 70s. We had gas rationing, so you'd either get your gas on an odd day or an even day, depending on your birthday, things like that. And there was quite a demand in the country to do something about renewable energy and finding alternative sources to fossil fuels. And that led to a series of years, about a dozen years, where we were funded by a bunch of agencies here at Harvard Branch in our old Aquaculture North complex to grow seaweeds for biofuel experimentation. We focused, after a lot of species screening, we focused on two groups, Gracilaria, multiple species, and also sea lettuce, the ulva. Both of them are very productive. These are much higher yields than you find for terrestrial crops, for example. They grow year-round. A lot of terrestrial crops are seasonal. They only grow in the summer or a certain part of the year. And some of this was really the first time that we understood that macroalgae could grow really fast when conditions were right. We did a lot of work on things like light and temperature and nutrients and carbon dioxide, trying to determine how we can maximize or optimize growth. This is a slide that just shows the difference between night and day. This is dark versus light versus how fast they could take up ammonium, a nutrient. And what that shows you is that's a lot more efficiently done in the light than it is at dark. So if you're interested in nutrient recycling efficiency, you're going to fertilize in the light and not overnight. I actually did my own digesters. These are anaerobic digesters. Methane gas was the product, which is the major ingredient in natural gas. About 60% of what comes off the digester is, in fact, methane that you can use directly. And again, keeping in my theme of nutrient recycling, one of the things I was really interested in was then taking the waste, the residue, left behind this digestion, which is liquid and some solids, and then recycling it back to the Gracilaria cultures. And we basically closed the loop here. So we could grow the Gracilaria, put it in a digester, tap off the energy, methane, and then put back the nutrients and keep using the same nutrients over and over and over again. And that actually was quite a lot of fun to do that. So again, energy flows one way, but nutrients cycle. Since then, we've used seaweeds for a lot of other purposes. We have some people that we work with that use seaweeds, Gracilaria and some of the reds, to feed to aplysia for neurobiological research, because the The neurons of Aplysia are great models for our neurons, so it's quite a lab rat. And we've used this for diets in conch. We've used it in diets for ornamental fish, such as these clownfish. And we actually use seaweeds as they are for home aquaria use, because it's a pleasant aesthetic thing to have in an aquarium. And also it's very nutritious for the herbivores that are in the tank to eat seaweeds. So why did all these efforts on biofuels stop more than 20 years ago? Well, bottom line is always money. Unfavorable economics. Back then I'd calculate that growing seaweeds for this purpose was worth something like a penny a pound. And it's worth a lot more for food or for aquarium use and all kinds of other things. So energy prices were low in the United States. There was unfavorable political climate, limited interest in renewable energy. There was sort of a resistance to try something new and different. I think this was a lack of vision, because I think, just showing you that one graph about the baby boomer image I portrayed there. I mean, at that point we already knew where the amount of fossil fuel was heading. So we knew it was a long-term problem. It requires long-term solutions. It's not something you're going to fix in a year or two. So how has it changed since then? Well, this is prices of oil, barrel prices. And the work I'm talking to you back then was back here, when we had these oil embargoes back here. And you can see the price now is still very historically low back then. You know that over a couple of years ago, we had this huge spike in prices. And then it fell back. And it fell back because of primarily a global recession. The important thing here is the increase we saw in the last 10 years is due primarily to a sharp increase in demand. It's not the politics of the 70s. It's not interruption in the supply. It's primarily driven by increase in demand. It's because there are other countries in the world, specifically China and India, that have greatly increased their fossil fuel consumption because they want to do the same kind of things that we do. And there's a tremendous amount of cars and things that are in those countries now that 10 years ago weren't there. So it's just a global problem now. World oil reserves have peaked. This has peaked really in the late 80s. This appearance of an increase here is just because another category was added, which is Canadian oil sands. There's some serious problems with Canadian oil sands. You can see the 60 Minutes episode that was on last year if you want to know more about that. But basically, the amount of oil reserves is flat. Our oil production, we're using oil faster than we are discovering new oil reserves. The best estimates I've found is that our oil reserves are good for 40 years at current rates of production and discovery of new reserves. Okay, so it sounds like we got 40 years, but as you know, once that supply starts to drop, it's probably going to bring somewhat of a turmoil like we had a couple years ago. You've heard several talks in this series about global climate change, and you've probably seen this slide by other people probably 5 times. But you all know that the CO2 from all the burning of the fossil fuels has altered our global climate. Algae have great advantages over agricultural crops. They grow faster on a per unit time and area basis. They can grow around. They can readily be converted to methane. They can convert it to other objects, to other products like cellulosic ethanol. And most importantly to me, they don't impact food prices and availability. And as you know, this turned out to be a big problem that was, we realized sort of late in the game. With all of the effort in ethanol, that had quite a large increase in food prices. Might not matter to you or me too much in this country, but it matters hugely to some of the developing worlds. This is a serious problem. It's really not wise, in my opinion, to convert food supplies to energy supplies. It's worth a lot more for food. So there's been a renewed interest in microalgae in particular, and I'm sure you've heard about this. But maybe you didn't know that for 20 years, there was a program, and we were part of this in the early years here at Harbor Branch, on looking at microalgae for renewable transportation, biodiesel. You've probably heard that word a lot. It's not economically viable yet, but there's a lot of interest. There's a lot of money going into it. This is one of the systems where it's a rack. It's kind of a high-tech approach. And some of the yields that have been reported are quite high. And this is an artist's concession of what a farm like this in the desert might look like someday. The algae that are being used primarily for microalgae actually produce oil droplets. Here's some of them right here in this green alga, Botryococcus, or oil drops in this Diatom cyclotella. So there are certain microalgae that actually produce oil that pretty much can be used as is with a little bit of modification. When did I start to take this real seriously? Well, I started to take this real seriously a little bit before this ad, but when this came out, this article that Exxon was going to put $600 million into producing algal biofuels, and that this was with Craig Venter, the guy who basically genomics pioneer, human genome, etc. I said, okay, now we got to take this seriously. But when I really caught my attention, it was one night as I was walking out during a commercial at home on primetime NBC Thursday night. As I'm walking out the room, I said, did that guy just say he was growing algae for 35 years? No, for 30 years. I couldn't have heard that right. There's no way there's a commercial on about algae on TV, primetime. But I was wrong. We can darken the lights now.
I've been growing algae for 35 years. Most people try to get rid of algae, and we're trying to grow it. The algae are very beautiful. They come in blue or red, golden, green. Algae could be converted into biofuels that we could someday run our cars on. And using algae to form biofuels, we're not competing with the food supply. And they absorb CO2, so they help solve the greenhouse problem as well. We're making a big commitment to finding out just how much algae can help to meet the fuel demands of the world.
Well, $600 million, Exxon, Craig Venter. I should point out Joe Weissman, some of the folks in the room recognize him. He was at Harbor Branch for a number of years. He was on our campus growing microalgae to feed the clams. So he was a clam farmer using microalgae. He has an extensive career working with microalgae, so I was quite pleased to actually see that commercial. I think biofuels is one of many solutions that are being looked at. I think wind and solar are going to be there a lot quicker. I think we're going to convert over to a much more of an electric power grid for our automobiles. And some of you probably know that FAU is very interested in ocean energy, trying to tap in the energy from the Gulf Stream using turbines. And some of you have heard talks about that in the past as well. So I think there's some potential here. I think that I'm more interested myself in the macroscopic algae. That's what I've worked with. Remember, I came to solve the world's energy crisis 30 years ago. And one of my colleagues, Terry Chopin, who has been to Harbor Branch in the last couple of years, who had been a postdoc here many years ago, has really promoted the idea of what is now called integrated multi-trophic aquaculture. So you grow things like salmon in pens, and they are polluters. I mean, anytime you have animals, they're going to produce waste. We have shellfish that take up a lot of the particles. This is mussels. And then you can have seaweed. In this case, it's kelp, laminaria, that can take up the inorganic nutrients. So you are getting multiple crops in one area. You're doing it in a very environmentally benign mechanism. So to close up, I want to say, why are these algae unappreciated? You guys told me they were scummy and all that at the beginning, remember? I didn't forget that. I took your names down. I found this quote when I first started teaching a few years ago, and it goes something like this. Plants are the most important, least understood, and most taken for granted of all living things. We just assume that they're going to do what they do. We don't really stop to think about it, and that's why I tried to make you do today with the algae. And I think that even for algae, it's even more true because they live in the water. They live out of our daily view of the world. And I talked about all these phytoplankton in the ocean. I mean, that's far away to us, just like that poem I mentioned earlier. And, but now you know that without algae, we would not be here. And you know something about energy flowing and nutrient cycling. And I want to thank all the agencies that support any of this work. There's a photograph of John Ryder, again, who was the reason I came to Harbor Branch in the first place. And we do get some of our current work on the Inner Lagoon with the Save Our Sea specialty license plate. So I thank anybody who participates in that. And just, I think the last word, though, really should go back to Einstein, don't you? Not Walter this time, though. I think we should go back to Albert. And one of my favorite Albert Einstein quotes is, if we knew what it was we were doing, it would not be called research, would it? I think Albert knew what he was talking about. Thank you.